Hi, welcome to Neuroverse
a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
In today's episode, we're joined by Professor Sonja Hofer. Sonja Hofer is a group leader at the Sainsbury Wellcome Centre for Neural Circuits and Behaviour in London since 2019, and she undertook her PhD at the Max Planck Institute in Neurobiology in Germany, after which she joined UCL as a postdoc and a Wellcome Trust Research Career Development Fellow, and also from 2013 to 2017 held an assistant professorship at Biocentrum in Basel, Switzerland. Her lab's research aims to understand the synaptic organization, function, and plasticity of circuits underlying visual processing in the mammalian brain. And her lab is focused mainly on understanding the differences in contextual signals that are integrated with feed-forward visual informations, how such signals emerge, and also on the thalamico-cortical and cortical-cortical interactions, as well as decision-making. She's also been awarded an ERC starting grant and Candel Young Neuroscientist Prize. Welcome to the podcast, Sonja.
Hello, very excited to be here.
We're really happy to have you. So I guess I'll just start off with asking what drew you into neuroscience in the first place? What sparked that interest for you?
Yeah, it's a good question. I came from animal behavior. So I studied biology at university, and I was very interested in, for example, how weaver birds learn to build these very intricate nests, right? And also how animals behave and interact with their environment. How do they gather food? How do they decide how far to go, where to go, when to kind of exploit the resources they have or search for new information and new resources. And kind of it became clear relatively fast for me that to understand these processes and these kind of behaviors of animals, I need to basically go to the brain and look at the neural circuits underlying these behaviors, and then we can maybe understand how they shape behavior.
Yeah, makes sense. And did you have any specific inspirations, such as scientists or figures that inspired you into a specific area? So why systems neuroscience? Why not molecular neuroscience, for example?
Yeah, so it kind of has a lot to do with my career path, right? The mentors and supervisors I've been working with, the labs I've been working in, but I've just never been that interested in the kind of the really nitty-gritty of cellular biology, of which iron channels are exactly expressed where and how do they maybe change with plasticity, or how neurons integrate signals on a single cell level and so on. That's interesting too, but that was never my passion. And on the other side, also the more cognitive neuroscience, I never found it particularly satisfactory to, for example, an fMRI to see, an area lights up in a certain behavior and so on. That for me was kind of very far away from understanding the brain. So kind of circuits and systems neuroscience really sits in the middle there, right? I wanted to get into the nitty-gritty of which circuits, which cell types, how do neurons work together, how different brain areas work together to kind of mediate different brain functions.
I like how you put that, because on Neuroverse, we always discuss the relationship between structure and function, and how another important component is the dynamics between the structures that lead to the functions. And I think what you expressed was basically how it's more interesting to look at the dynamics of a system rather than just, this place lights up, it's responsible. It's not necessarily the case that means the region is responsible. It's more about the dynamics and the communication between regions. And that also really interests me and Carolina.
Yeah, exactly. I mean, if you go on the single cell level, right, neurons are pretty well understood. So there's not much magic left to discover in a way, right? But then the magic comes when you get to a huge complicated system of neurons interacting and the dynamics between them, right? And this is where we lack still a lot of understanding, I would say.
Yeah, that was actually going to be one of my questions, in that some labs, they specialize in specific brain areas, such as the medial internal cortex or the hippocampus. And I feel like your lab is always focused more on the circuitry and between areas, thalamical cortical and cortical cortical. So, but it's also important to understand the specific functions each brain area has to know where to start. So how do you balance that? How do you balance understanding the function of a specific brain area or system, and then perhaps more higher level stuff and connecting the dots? Like with the IBL paper that recently came out about priors and how it confirms it's more according to the Bayesian model and not, specifically priors being encoded in one brain area.
Yeah, so it's interesting because how I started was very much area centric, right? It was very much in primary visual cortex of mice and looking how different neurons there were connected, which different cell types, how they talk to each other, and how do the circuits change during learning. So we kind of started on the local and single area level. And I mean, that's what lots of neuroscience labs have done for many decades, right? But now I think we're really getting more and more away from this notion of one area is having one function and another area is having a different function, right? It's very clear that a lot of brain functions are very distributed and involve many, many different areas. And both in the neocortex, where a lot of the research was focused in the past, but also in kind of tiny subcortical nuclei and so on. So I think that's kind of where neuroscience is going more and more, is really looking at these distributed circuits, looking more and more brain-wide, and rather than saying, okay, I have to study now only hippocampus if I want to understand memory.
Yeah, I think particularly with learning and memory, this is a really kind of a shift in the field of neuroscience, I guess, because it used to be so hippocampal centric because that's where LTP was discovered and everything. But then there's actually so much plasticity that occurs in the cortex, which is amazing because then you can study all of the sensory processing, learning, and memory, which I guess is what you do.
Yeah, speaking about the cortex, so really exciting. Shohei, who's a postdoc in Sonja's lab, his preprint came out, I think yesterday, about predictive coding and the thalamical cortical circuitry. And as we all know in Neuroverse, predictive coding is one of my most passionate topics. So I'm really excited that I get to talk about that with you, Sonja. So could you first explain what predictive coding is and what the predictive error signal is?
Okay, let's maybe start very wide.
Okay, yeah.
What my lab is really interested in, and what we kind of know intuitively and from many optical illusions and so on, is that what we perceive from our environment is not just what we passively take in through our sense organs, through the eye, for example, right? So it very much depends, or is modulated by our knowledge, our expectations, our intentions, kind of really internal information. What do we know about the world, and what do we expect, for example, in a specific context, or how something might look, like how a house might look, and therefore we might interpret something that we see in the environment as a house. So a lot really depends on what we call our internal models of the world. And predictive processing is kind of one sub-model of this framework that says we have internal models, and from these we have expectations or predictions that could be sent down to our sensory processing areas and are somehow integrated with a feed-forward sensory input. And that allows us to really interpret, for example, visual information that's coming in, and that's called inference, right? We have to, from the often ambiguous or partial information we get on our retinas, these pixelated images, we need to somehow guess what's out there. What are we actually seeing? And that's, so we're inferring our environment from this information. And predictive processing is kind of a subgroup of this framework that says there are these very strong prediction signals that coming top down from a higher cortical area to a lower one, that are then in this lower area compared with, for example, the visual input coming in, and see if they match. And if they match, they're canceled out. And the only thing that's sent up further up is the so-called prediction error. So if there's a deviation between the predictions and the actual visual input coming in. And we basically wanted to look both at these so-called prediction errors, what is happening when our expectations are violated, for example, and more general, where are these internal models actually stored in the brain, and how are these prediction signals look like?
And so what did you discover in terms of where do you think that the prediction signals are originating from or projecting to, and I guess, like you said, where the internal model is potentially stored?
Yeah, so I think that's a very big question, and we're really just scratching at the surface there. So we're nowhere yet. The Shohei's paper really nicely, I think, describes the prediction error signals we see in primary visual cortex. So other papers and other studies have described that too, that, as I said, if we perceive unexpected sensory information, if we see a stimulus that we're not expecting, then there are these enhanced signals in even kind of lower sensory areas that we get these stronger responses in response to unexpected information. So that has been described in different contexts and for different kind of prediction errors, but it was still very unclear what is actually the nature of these signals. What do they actually encode? What do they represent? And what Shohei found is, so first of all, the prediction error he described were, I would call them maybe spatio-visual prediction errors. So in an environment that we put our animals in, so it's mice that are traveling through a virtual environment, through a corridor, and they see different visual images on the wall. And mice learn kind of the sequence of these images and learn to expect specific images at locations in this virtual environment. And then we can play with their expectations. We can violate them. So, for example, show them a new image at a particular locations or swap images around and so on. And then we see these prediction error signals. And what Shohei found is that these prediction errors, at least their kind of dominant feature, is not they don't encode the difference between what's expected and what's the actual visual input. So this is one thing that most predictive coding theories hypothesize, that these prediction errors should give information about how the actual information deviates or is different from what we're expecting. And this is not what we found or what Shohei found. He found that it's really what's represented is the actual visual information that's unexpected. And that's very specific. It's also not just a general surprise signal that the animal is maybe twitching because it's surprised or something. So it's not a motor signal. It's not a general signal. It's very selectively the information that is unexpected is basically boosted and their salience is increased. So probably to allow downstream areas that read out this information to prioritize it, right? Because now that's particularly salient.
Yeah, that's fascinating. Why is that insight in that the predictive error signal may not be the difference in expected information, but more the unexpected information? Why is that distinction important in terms of relevance for updating the internal model?
Yeah, I mean, as you said, when thinking about what these prediction errors are good for, what is their function? One main theory is that they are used to update the internal model and update the predictions that we have. And depending on what is the nature of these signals, we can then build models of how this updating would actually work. And do we actually need to know what's the difference between what we expect and what is the actual to update them or not? To know the details of this will just help us to actually build models that are maybe more realistic.
So that's fascinating. From a more like energetic point of view, so if we kind of view the brain as trying to always reduce the amount of energy expenditure, the fact that the novelty or the novel stimuli or the unexpected stimuli are more salient and lead to a larger signal, could it be that the point of the brain is to essentially then reduce those signals and constantly adjust to new unexpected stimuli? And how would that overcome? Would that then mean that you need to always have the same unexpected stimulus to then make it familiar? If you only have a new stimulus once, then you'll never kind of adapt to it. But then if you have the same one over time, then is that sort of how it's thought to work?
Yeah, no, but exactly, that's exactly how we think it works, that if you then encounter this novel stimulus more often, right, you learn to expect it in this situation. So then this prediction error signal is gradually going down as you update your expectations. So so what we think is that maybe stimuli that we expect, that we're used to, will evoke much lower activity in the brain than things that we don't expect or that are novel. And then over time, their activity will go down again. So that's one thing, is that this will reduce the energy needed, but it also allows the brain to kind of prioritize this novel information, which many times might be the most relevant one. This is the one I want to know more about, and if this doesn't match what I'm expecting about the world.
What do you think are the consequences if this prediction error signal doesn't occur properly? So, for example, I'm aware that perhaps autism and schizophrenia are sometimes used as models to investigate top-down versus bottom-up processes. Can you speculate on what do you think is the consequence if this prediction error process doesn't occur properly?
Yeah, so I think the whole system, when it goes awry, what happens then is very interesting. So one theory for schizophrenia is that these patients can't really distinguish what is real and what is imagined, or what are kind of our top-down, maybe prediction signals, what comes from inside me versus what is outside, right? And that's maybe even a level higher than the prediction error signals. Can I distinguish my internal processes from the outside? And then one would expect, if that's the case, then also prediction errors to be faulty or not be represented properly, right? So yeah, at the moment, I think really kind of compelling evidence for this theory is still lacking, but that's really a super interesting avenue to pursue.
Yeah, absolutely. A lot of clinical applications potentially. And so in this paper, you also spoke about the mechanisms underlying this prediction error signal and how it has this complex inhibitory circuitry, and that the VIP cells inhibit the SOM cells, which are both inhibitory interneurons. Could you pick out the circuitry a little bit and explain a little bit the mechanism, please?
Yes, I go into the nitty-gritty details.
Yeah.
So first we found that the thalamus is kind of important for these prediction signals. We have like a pet area that we're particularly interested in the thalamus, which is called the pulvinar, or in mice also the lateral posterior nucleus. But I think it's now established that that's a homologue of the pulvinar. And the pulvinar is one of the largest thalamic areas in humans. It kind of grew together with the cortex during evolution. And it's a visual area, but kind of a higher order thalamic area. So in contrast to places like the LGN, the lateral geniculate nucleus, which is a first order thalamic area that is kind of the relay from the sense organs to the cortex. And we've been very interested in kind of the function of this pulvinar, these higher order circuits, and how they interact with the cerebral cortex, because they're very densely interconnected. And cortical processing kind of very much depends on these thalamocortical circuits. So what we found is that thalamic input is important for boosting activity in cortex as a response to these prediction errors. But we also found that under normal circumstances, this input is kind of gated in the cortex by a particularly type of inhibitory interneurons, the somatostatin expressing interneurons. So normally, if information is coming in from this thalamic areas, there's a lot of inhibition from these somatostatin cells that kind of suppresses their influence. But there are these interesting disinhibitory circuits, as you said, inhibitory circuits in the cortex, that there are other inhibitory interneurons, the so-called VIP cells, and their main target are these somatostatin cells. So they can inhibit those and therefore disinhibit the excitatory cells that the somatostatin cells target. So you have kind of a disinhibitory mechanism. So if these VIP cells are active at the same time as the thalamic input, then they kind of have this synergistic effect that then the gate is opened and thalamic input is then allowed to have an influence on cortical processing and have an excitatory effect, and then can boost responses in the cortex. And we see that it's doing that in a very selective way.
Interesting. And so what happens when there is a prediction error signal? So that allows the thalamus to then be able to excite the cortex?
Yeah, so we found this interesting, a little bit of a conundrum, that we see that there's basically two streams of information that need to reach the primary visual cortex. One is through the thalamus, and another source, or it might be the same source, also needs to impinge on these VIP cells in the cortex. And both need to be active at the same time, but independently. So that's still a completely open question for us. What is this input that impinges on these two main players? And is it different information, or is it the same? Is it simply kind of a mechanism that allows very specific and efficient boosting? So my hypothesis, my pet hypothesis at the moment would be that the prediction error is coming through the thalamus, or is at least boosted through kind of thalamocortical circuits, these loops, and the VIP cells maybe receive more general signal that something is surprising or something is particularly behaviorally relevant, because VIP cells also respond to other events that are very salient, like receiving a reward or receiving punishment or novelty of a stimulus. So that's maybe a more general signal that then allows the thalamus to have an influence and say, okay, boost the responses to this stimulus because we think that's particularly relevant.
Interesting.
To get even more molecular, do you hypothesize any different neurotransmitter systems? Of course, the VIP and SOMs are GABAergic, but do you think with the salience that a different neurotransmitter would lead to these activation of the interneurons?
Yes, that's a very good question. So one theory would definitely be that the VIP cells might be activated by some neuromodulatory system. So cholinergic input, or what we think is maybe more likely is noradrenergic input. There's been a recent very nice paper from the Georg Keller lab. That's a lab that also is very well known for looking at the mechanism of predictive processing. And they found that noradrenergic neurons seem to convey a different type of prediction error than we have discovered, but seem to signal prediction errors. So it could be that the VIP cells are boosted by these noradrenergic signals.
Fascinating.
Yeah, really interesting. I also wanted to ask that, so this is all focused on the primary visual cortex and the signals that project there. Do you think all the different areas of the thalamus have their own prediction error signals that lead to different cortical regions? And do you think there's multiple different types of prediction error signals across the brain?
Yeah, so there's definitely many different types of prediction errors, right? So the one we now looked at, this paper was really this kind of more spatial prediction error that we think maybe involves the hippocampus, maybe retrospinal cortex, right? It's kind of knowing my environment and what to expect at a specific location. For that, probably, and for navigation in general, these hippocampal, maybe retrospinal cortex circuits are probably very relevant. Then another type of prediction error is more the sensory motor prediction error. So given that I do a particular motor action, for example, moving my eyes, that also causes a sensory input, sensory feedback, right? So for example, it shifts the world on my retina if I move my eye. So also there's strong internal predictions of what is the consequence of my actions. If that doesn't match, if the feedback I'm receiving in response to a motor action I'm doing is different than what I'm expected, then that's a different type of prediction error, like for example, a visual motor prediction error. And these would definitely expect to be computed and processed in different areas. So maybe more sensory motor area, like there's some indication that maybe the secondary motor cortex is important for that, and maybe also the superior colliculus, and then pulvinar could convey these signals from both and integrate them and get them back to the cortex. One big question for us is kind of where is really the error computed? Is that in V1 or in the thalamus or somewhere higher up the hierarchy? And my guess would be it's higher up in the hierarchy and maybe more something like prefrontal cortex, or in the case of these spatial prediction errors, maybe retrospinal cortex, who knows? And that is conveyed back to visual cortex and then maybe amplified in this visual cortex thalamus loop.
So when you were talking about the different types of predictive error signals and potentially this one in the motor cortex, it reminded me potentially of the cerebellum and how there's some research about prediction error in the cerebellum. Could you explain how that's different from the prediction error that we see in the cortex, or if it's a similar type of signal?
Yeah, I think in principle, it's a similar type of signal, right? And it's interesting there, it's very clear these error signals in the cerebellum exist, right? They're important for learning, for again, updating my motor plan. And maybe if I want to do especially very intricate, very complex movements, then prediction errors are probably important for telling me, oh, this was slightly wrong, do it slightly differently, and so on. So it's more for motor learning, versus these sensory prediction errors are maybe more about updating my sensory models or kind of the representation of the world in my cortical circuits. But in principle, it's, I would say it's the same principle. And it's interesting that even in the cerebellum, where these prediction errors have been researched for much longer, we still don't really understand how they're computed, right? So I think it's a bigger question. The same in the basal ganglia, where that's kind of the most prominent or well-known circuits for prediction errors, for these reward prediction errors in dopamine, in the dopamine signal in the striatum. And even there, it's still unclear where and how it's computed.
So you mentioned behavior and motor output quite a few times. And so I just wanted to ask, in terms of prediction error signals, how relevant are they for the behavior of an animal? With the unexpected stimuli, you mentioned being attracted to it, but we also want to avoid them, so that fits our internal model. How is this kind of balance for behavior?
Yeah, that's a super interesting question that we're also really interested in. So we think that animals are actually really attracted by this uncertainty. So if it's something they can't explain or don't know, or also us humans, right? This is something we want to explore further. And many animals have this really a strong drive, its own motivation of gather new information to explore the environment, which makes sense because it's super important for survival. If I don't know about new food sources in my environment, if I don't find a partner somewhere, any mate, then it's not good for my survival or the long-term survival of my species. So I think there's really this urge to explore and especially find out more about things that are uncertain. And this is something we really in the future want to study, the next step of if there's prediction errors, if there's something I can't explain, if there's no pattern I can or sense I can make of this new information that I'm getting, is this really attracting animals to explore more, and through which mechanisms? So where does this motivation to explore come from?
Yeah, and of course you study mice. As you mentioned in the beginning, your interest in biology and different species of animals. And we also just recorded an episode all about different, more unique animal models in neuroscience. Because of the relevance of this prediction mechanism for exploration, do you think it's something that occurs across many species, or is it more in higher order species and mammals? Or what is your, of course, I don't think this is really known, but what is your sort of hypothesis about this?
Yes. I haven't really thought about where would I think evolutionary this error signal come from. I mean, obviously, exploratory drive is really something that basically all animals do, right? Yeah, I need to find new food sources and something, or even kind of paramecia or something. It'll have to explore its environment. But probably I wouldn't expect prediction errors in these very simple organisms. But I don't know, it might actually be, right? That I'm expecting something sweet here, but actually it's sour. I should go somewhere else therefore, or something like that. But yeah, it's probably simpler in that way. But I would certainly think all vertebrates to have them. And who knows, yeah, maybe also higher invertebrates like cephalopods. I mean, it's certainly possible, right? It seems like such a fundamental way of learning that I would expect that in many different animal kingdoms and systems.
Related to exploration, you also carry out research related to fear regulation. And so how do you think that those two are tied? And potentially, do you think that VLGN plays a role in this?
Yeah, so this is kind of a new direction in the lab that we are going more and more into. I mean, it's tied to my previous research that is more kind of sensory processing and perception, in a way that we're very interested in how these sensory signals are integrated with internal information. And on the one hand, for perception and how we perceive the world, and then on the other hand also to guide our behavior, right? So for the decisions we make, we take into account many things, the present sensory information, but also previous experience, our knowledge, our internal state, all modulate how we will decide. So one avenue of research we're pursuing at the moment is how this kind of integration of these different types of information can regulate fear behavior and anxiety in animals. And there we found this new and unexpected hub that can integrate a lot of different information, and that seems to be really important for the control of fear reactions and also for kind of more general anxiety-like behavior in mice. As one always has to be obviously a bit careful when talking about things like anxiety or other kind of mental states in animals, because we of course can't ask them, are you anxious? But we can measure their behavior, and so one needs to be a bit careful to maybe not call it mice are anxious, but mice might show anxiety resembling behavior.
Yeah.
And we know that this pathway, this so-called ventral lateral geniculate nucleus, the VLGN, is an important hub that can control these types of behavior.
And could you tell us a little bit about some of the research you found, for example, like the paper with Alex?
Yeah, so we found that this VLGN can kind of bidirectionally regulate fear responses. So for example, if we expose animals to a stimulus that they perceive as threatening, so we show them, for example, these black looming stimuli, so expanding black discs, which we think mimics a predator swooping in from above. And that's a very innately threatening stimulus to mice and to many mammals, actually to many animals, not only mammals. And that induces these fear reactions in mice, either kind of an escape to a shelter if they know that there's a safe place somewhere, or if there's not, then they would freeze, which kind of just be very motionless to maybe hope that if there's a bird over over them that they can't see them anymore.
Yeah.
And we saw that these fear reactions can be very strongly controlled through this VLGN, this ventrolateral geniculate nucleus. So if we, with, for example, optogenetic methods, we can switch on and off specific cell types in this nucleus. And if we switch them on, and this nucleus is mostly inhibitory. So if we increase the activity of these inhibitory cells, then we can suppress very strongly these fear reactions. And also vice versa, if we decrease activity in these circuits, we can make mice more fearful, and then they respond stronger or to more subtle threatening stimuli, and also show generally more anxiety-like behavior. And we also found that activity, at least in a subset of cells in the VLGN, is modulated by the experience of these animals and their expectation of threats in the environment. So we think it can regulate these fear behaviors depending on the animal's experiences, its knowledge, and probably also its internal state, how anxious it is in general. So it receives input from many areas that might bring these types of information.
Yeah, it's interesting how it all is so context dependent, and it ties into, I think, the importance of when studying a circuitry and, you know, a specific question, to look at all of the different brain areas connecting and to look at all of the variables present, like the different contexts. You mentioned earlier, you know, we need to be careful with our language when describing their behaviors and what we are observing, such as anxiety-like behavior, because we can't ask them how they're feeling. And so this ties into my next question, which is, what do you think are the main challenges and limitations in studying sense perception and internal states? So there's this one in which, you know, we can't ask them, but in terms of also, you know, technological challenges and potentially anatomical challenges, for example, the striatum and separating the two different populations. So what for you do you think are the main challenges and limitations?
Yeah, I mean, in terms of sensory perception, I mean, of course, in these animals, we can't really study perception, right? Because we don't even know how they perceive the environment in the end, right? So it's really more the sensory processing of the information we're studying. And we can read that out as a function of the neural dynamics in these different areas. But questions like how are different aspects of a sensory input combined and bound together to lead to our perception, that is very hard to study in animals. As a readout, we then use behavior. We don't use their perception. We kind of ask them, have you perceived this stimulus, or is this stimulus different to another stimulus? And they need to do a motor action to tell us this, right? So with this, we can find out maybe what they have detected, or if it's been different than another stimulus, but it's indirect, right? We can't directly ask them, what are you seeing?
Yeah.
So that's definitely the main challenge, I would say. And that's the same thing as I already mentioned with looking at internal states and things like fear or hunger is easier, right? Because we can, if we take away their food, we know like a few hours later they will be hungry. So it's again a readout of behavior and things like fear and anxiety, that we can see how do they behave, and does that tell us from previous studies that they're more or less anxious. And so that we have to use these indirect measures is, I would say, the biggest challenge. Otherwise, methods-wise, I mean, nowadays we have so many really sophisticated methods to switch on and off activity in very specific circuits, to target very specific pathways and cell types. And every week there's a new, really exciting tool that we can use. So I think there, in that respect, there's fewer challenges. I mean, there's challenges, but ones that we can overcome, certainly in the next decades or so.
Amazing. So what do you think are the next big questions in neuroscience that we don't have the answer to, in your opinion?
I think there's too many of them to pick one. I mean, one for me, I think is really very much related to these predictions and prediction errors, that we still really don't know how our internal expectations, predictions are integrated with the sensory information coming in, and how that leads to our perception, right? So where and how this is done, I think, is for me one very big question. And then also, I mean, there's many others, like how do we really learn? How is new information integrated in the very distributed circuits of the brain? How is it stored? How can it then be retrieved to be used for behavior? Is another one, but I could probably come up with 10 or 20.
Yeah, of course.
Still so much unknown.
But what do you think would be the main way that we could tackle these questions? So, for example, we can try and train AI based on neural networks, and how and try to see if artificial neural networks can learn in a similar way that brain networks learn, and we're still quite far off. Do you think that using computational models is the way forward? Do you think we need more translation between animal models and humans? What do you think would be the key things what would be the gaps that need to be bridged?
Yeah, I think one thing is really definitely theory and models, like finding the algorithms that we think are implemented by specific circuits. I mean, just being able to record from everything, but without theories to address, is very hard to lead to really knowledge. So if you just go to a brain areas, let's say the posterior parietal cortex or something, and we just can record from all the neurons in different contexts and so on, that still probably won't tell us what is the function of this cortical area, right? So what we need is really ask, what are the algorithms that could be performed by specific circuits? What are my theories of what these circuits could do? And then design very specific experiments to test these theories and be able to validate or invalidate them. I think that's one very, very important aspect that's of course there, but I think should be even much bigger. So the collaboration between theorists and experimentalists should be much closer. And then if we have that, then I think going forward, then it can be very informative to be able to, there's very exciting methods out there, and they're being developed further and further all the time to record from many neurons at the same time. But we want is also know their cell types, maybe their projection, where do they project to, where do they get their input from? And if we can combine all this information, and then also obviously being able to manipulate very specific circuits and cell types while recording from many places in the rest of the brain. Yeah, that's, I think, then gets very powerful.
Yeah, amazing. I have one more question. So one thing that I admire about your research and the discussions that we have in lab meetings is you have an amazing way of seeing the logic and seeing the flaws in someone's thought pattern. No, but I say that in the best way possible. I think it's amazing, and I really strive to have a bit more of that way of understanding the logic, and when things make sense and when they don't make sense, and the best way to find the solution. What advice do you have in terms of like the type of scientific method to have and how to approach problems? And yeah.
Yeah, that's a hard question. But yeah, I think just thinking deeply about things and trying to get to a problem from many different directions is definitely important. But asking yourself again and again, also, if you have a result, can I explain this in any other way as my theory? How can I rule out that there is some kind of artifact or something. Just questioning yourself all the time and your experiments and your knowledge also, right? To be able to, if something changes, if there's new evidence, you need to be flexible enough to actually adapt and say, yeah, fair enough, I was wrong. You're right.
Yeah.
I think the older you get, often that's not as easy anymore. Then you can get too fixed in your beliefs and so on, and that's obviously very dangerous.
Yes, of
That's
course.
why it's important to have the new generation with new exciting views.
Yeah.
That's great advice for us young neuroscientists.
Yes, I'll definitely take that into
yeah, ask
account.
exciting questions and
Mhm.
go crazy, and don't be too restricted by the old guys who think they know everything.
Amazing. I love that. Yes. Okay, I think this is a perfect place to end today's episode. Thank you for joining us, Sonja. It's been a pleasure to have you on.
Yeah, it was a great pleasure. Thanks for the very interesting discussions.
Yeah, thank you so much for sharing all your knowledge. This episode was made possible thanks to the support of the Sainsbury Wellcome Centre's Public Engagement Fund. We'd like to thank the SWC once again for their support in promoting important conversations like these.
Yes, we're really looking forward to recording more episodes with people from the Sainsbury Wellcome Centre at UCL, as well as the Gatsby. And yeah, if you like this episode, please feel free to share it on Twitter, Instagram, and we have links to any papers and resources that we used for all of our conversations on the Neuroverse website.
Thank you for listening.